So hello guys and welcome back to Accessory Academy. So from now onwards, we'll be starting with the repair and a different mechanism by which the DNA which has made mistakes. Okay, if suppose any mistake is being done during the replication, that will be corrected. It's not only about the replication error only, but also like that many other alkylating agents are there, right? So those agents can also cause DNA damage, right? So those kinds of damage, how to repair? If suppose there is a UV radiation or any other ionization radiation is there, then how DNA is going to repair itself, okay? With the help of the other repairing proteins before it is going for the mitosis. Okay, so this is important. So let's talk about the DNA damage. What are the different types of the DNA damage here? So it undergoes damage simultaneously from hydrolysis and deamination. Okay, it is spontaneous. So these are the spontaneous damage which can occur. So your even water is also going. Sometimes it can result in the spontaneous damage, which can result in the hydrolysis. Okay. Okay, so mutations arise not only from errors in replication, but also from damage to the DNA. It is caused by environmental factors such as radiations, which we know that there are certain chemicals called mutagens, okay, that can increase the mutation frequency. Now, it also undergoes spontaneous damage because of the action of the water. Now, let's see what are these different types of spontaneous damage which can occur to the DNA. So the most frequent one is the hydrolytic damage, which is also the deamination of the base of cytosine. Now, what do you mean by deamination, guys? Deamination means you are going to remove the amino group. Okay. So now if you see in this case, this is the structure of the cytosine base. You can see that you have got the amino group over here. Now, deamination means it is the hydrolysis, right? Water is being involved. Now what is happening? So suppose deamination of cytosine is happening, then what will happen? This is getting converted into uracil. Now why uracil is being considered a DNA damage? Even though we know that uracil is actually a natural occurring base, then why it is called DNA damage? Yes, Shruti?
Because it's not used in DNA, it's only used in RNA, so the structure will
Yes,
vary.
yes. So because of that, it is unnatural. It is unnatural for uracil to present in the DNA. Okay, so we know that like how A is making hydrogen bonds with T, it can also make with U. Okay, so uracil preferentially pairs with adenine and thus introduces that base in the opposite strand. So what will happen? See, अब आपको मालूम है DNA में if C is there, so in the next replication round, G should come in the daughter strand. But if because of this hydrolytic DNA damage, the C has become U. So what will happen in the next? It is going to give you A. So this is a error, right? So this is going to create sequence change. And that's why it is known as a DNA damage. And plus, uracil shouldn't be present in the DNA. And adenine and guanine are also subject to spontaneous deamination because even they have got the amino group present in them. Okay, so now deamination of your guanine and adenine. So what will happen? So deamination converts adenine to hypoxanthine. Guys, please, this you need to remember. So adenine to hypoxanthine, which can hydrogen bond to cytosine rather than thymine. So we know A is with the T, but A agar आपका hypoxanthine बन गया, if it is hypoxanthine, then it can base pair with C. So again, instead of having AT, next round of replication, what will it will happen? It becomes CG, correct? And guanine is converted to xanthine. So you are going to get xanthine in case of G, which can pair with C along with only 2 hydrogen bonds. Now even that is wrong, right? क्यों गलत है? Because for a stable structure in DNA, you should have 3 hydrogen bonds between C and G. But here it is forming only 2. It means what? This is not going to be a stable base pair in the DNA. Okay, so here you can see the hydrolysis is happening and this results in the deamination of guanine, which is going to create apurinic deoxyribose. So what does it mean? It means only sugar and phosphate group is present. There is no base. Okay, so that becomes apurinic. Similarly, if suppose pyridine was present and it was removed, then it becomes apyrimidic deoxyribose. Guys, this is clear? This slide is clear to you?
Yes, ma'am.
Yeah. Okay. So please remember, adenine gets converted to hypoxanthine during deamination. Guanine gets converted to xanthine during deamination. So these two cases are for deamination. Okay. And hypoxanthine can base pair with C, xanthine can base pair with C, but only with 2 hydrogen bonds instead of 3. So again, it is unstable. Now coming to the hydrolysis, in the case of hydrolysis, what will happen? A purinic or a pyrimidic site will be created. Okay, this is hydrolytic damage. Okay. Now, now hazard of having deamination, it generates a naturally occurring base. Okay. Now this is shown by the presence of the 5-methyl cytosine. Now suppose you have got the 5-methyl cytosine and it is undergoing deamination. What will happen then? So that 5-methyl cytosine is going to get converted into thymine. Now, can you tell me where can you find 5-methyl cytosine much? Where do you find methyl cytosine, methylated cytosine? In DNA, where do you find these? Yes. Anyone? Akshay, any idea?
No.
No. Have you heard of CPG island?
Yes, ma'am.
Where do you have, where does it come? The CPG island. What does it mean, CPG island? क्या है ये?
A series, an area where there are a lot of CG clusters. There's a lot of.
Yes. So in those CPG islands, you mainly see a methylated cytosine as well over there. Okay. Now, what will happen if suppose methylated cytosine is undergoing deamination? So we have seen in the structure now that the methylated cytosine under hydrolysis or deamination, it will result in the formation of the thymine. Okay. Now, what is happening? What is happening then? Again, it is going to create a DNA damage, right? That from cytosine हमें यहाँ पे क्या मिल गया है? Thymine. अब यानी कि CG base pairing के जगह we are going to have TA base pairing, right? Now, if you see, always remember this methylated cytosine is mainly in the case of the transcriptional genes. Okay, so they are the rich region present over there. And now you know, if suppose you have got the genes and वहाँ पे अगर ऐसे mutations हो गए, okay, like you have got C, G का होना चाहिए, वहाँ पे आपको T, A हो रहा है. So what is happening? At the gene level, the sequence is getting changed, right? So this is going to affect the transcription. So this is why it is important to understand this kind of DNA damage. Okay. Now, vertebrate DNA frequently contains 5-methyl cytosine because of the action of methyl transferase. Okay, so they have got role in transcriptional silencing. Now, deamination of 5-methyl cytosine generates thymine. Now, the problem is that the thymine will not be considered as abnormal, right? So it is going to replicate. Now, this is resulting in the transition. Now, if you see any DNA change, base pair change from purine to purine is transition, okay? Pyrimidine to pyrimidine is transition. But if it is purine to pyrimidine or pyrimidine to purine, then it is known as transversion. Okay, anyways, it is going to come in future slides, but again, try to remember, okay? Okay, so these methylated cytosines are the hotspots for the spontaneous mutations in the vertebrate DNA. So what does it mean, hotspots or spontaneous mutations? It means wherever genes are present, you know, the transcriptional active genes are present, the chances of mutation are also high. And that's why it is going to affect a lot, right, to the cell, to the organism. If there is a mutation in the transcriptional gene, it is going to give the wrong product. Either it is the wrong or truncated RNA or the truncated protein. It is eventually going to affect the cell function. So it is important that these kinds of DNA damage are repaired as soon as possible. Okay. Now you have got DNA damage by alkylation, oxidation, and radiation as well. So it is vulnerable to the damage from alkylation, oxidation, and radiation. So in alkylation, methyl or ethyl groups are transferred to the reactive sites on the bases and also to the phosphates in the DNA backbone. Now this is something which you need to understand. कोई भी methyl ethyl group हो गया, CH3 हो गया, C2H5 groups हो गया, ठीक है? So they are transferred to the reactive sites of the bases. Now for example, alkylating chemicals include nitrosamines, and the very potent one is the N-methyl, N-nitro-nitrosoguanidine. Okay, this is like a poison only. Please remember. So one of the most vulnerable sites of alkylation is a keto group at the 6th carbon of guanine. So what will happen? So this methyl is going to result in the O6 methyl guanine, which often mispairs with thymine. So it is a guanine. So instead of GC base pairing, I'm going to have the GT, right? So in future, what will happen after the replication? Instead of GC, that sequence is going to contain AT. Okay, is this clear to you?
Yes, ma'am.
Okay.
Yes, ma'am.
Please remember, huh? So we have just talked about one of the alkylating agent, जिसका नाम है nitrosamine, or in like the full name is also given. If you could remember this much is also fine, right? Nitrosamine सुन के ऐसा नहीं लगना चाहिए that you're talking about the amino group and all. We are talking about the methylation which is happening here. Okay. Now look at over here. So this is also important. So you see the structure of the G. Okay, guanine का ये structure है. So you can see where all kinds of the DNA damage can happen. So you have got this amino group which can result in the deamination, that removal of the amino group. Then these are the sites which can result in the alkylation. N, O, and again this N, and this is like जहाँ पे भी double bond है, there oxidation can happen. Okay, so these are the modifications which are like often highly mutagenic. Similarly, in the case of the adenine also. So DNA is also subject to attack from reactive oxygen species like O2 minus, H2O2, and OH radical. So these potent oxidizing agents are generated by ionizing radiations, which will result in the free radicals. And हमें मालूम है जो free radicals form होते हैं तो क्या होता है? So they might bind to they might affect the DNA structure or the proteins which are involved in the DNA replication. So they are going to affect the function of the cell, correct? So oxidation of guanine, for example, generates, this is something which you have to by heart, guys, okay? No need to remember the structure, no need of that, but please remember what is oxo-G. Oxo-G अगर present है तो किसके साथ base pair करेगा? वो translation में होगा या transversion होगा? Okay, so these kinds of questions they have already asked, so आपको इसका या फिर make a short note or whatever which is comfortable for you guys to remember. So oxidation of guanine. So we saw like where the oxidation can affect, where we have got the double bond, right? So it generates 7,8-dihydro-8-oxoguanine, which is in short is also known as oxo-G. Okay. Now, this oxo-G is known as adduct, is highly mutagenic because it can base pair with adenine as well as cytosine. See, if it base pairs with adenine during replication, see, अगर G का C के साथ हुआ है, no problem. But if it is base pairing with adenine, okay, oxo G is base pairing with adenine, then what will happen? Instead of GC, in the next round of replication, I'm going to have TA. What does it mean? It means then G से T हुआ, purine से pyrimidine हुआ, right? So this kind of mutations are known as transversion, and these are common mutations which is found in the human cancers. Okay, is it clear, guys? Clear? So you have got the reactive oxygen species, which can result in the formation of such addict. One example is oxo-G, which is also known as 7-A-dihydro-8 oxoguanine, or in short, oxo-G. Oxo-G can base pair with A as well as T, right? Sorry, A as well as C. C के साथ किया तो कोई प्रॉब्लम नहीं, A के साथ किया तो ट्रांसवर्शन होगा, ट्रांसवर्शन होगा तो मतलब कैंसर होगा. Simple. Am I going fast?
No, ma'am. Please.
Okay. Like many things are there to remember, that's all. Understanding is easy, but yeah, many things are there. Now, another type of damage is the ultraviolet. Okay, so we know that even we are going to visualize a DNA at 260 nanometer, right? जब भी हम किसी spectrophoto में check करते हैं, you must have heard of nano drop also. सुना है क्या कभी nano drop? Or spectrophotometer? Have you used in the lab nano drop and spectrophotometer? So nanodrop is like spectrophotometer, at least 1 ml of solution is required in order to check, okay, the absorbance. So mainly spectrophotometer is used for your protein samples, but you can also check DNA. Of course, you just need to change the wavelength. So we know that for DNA, it is 260 nanometer, right? Because that is the strongly absorbed by the bases. Now, if suppose I'm using nanodrop, don't worry, I'll be taking all these instruments. So if you're using nanodrop, from the name itself, you can understand that we are taking hardly 1 microliter. Okay, so here it is 1 ml, here it is 1 microliter. So can you see it is 10 raised to minus 6 of a liter, or I can say 10 raised to minus 3 of a ml, right? So how much less sample solution I'm taking just to check the concentration of DNA. So suppose I get 1 microliter of, DNA concentration as 500 or 560 nanogram per microliter. Okay, this is how you get in the machine, the absorbance, okay, at 260 nanometer. आप machine में read करोगे. अगर मेरे पास इतना है, then you can back calculate like how much I have in 20 microliter. You can't have DNA in 1 ml, okay? हम इतना ज़्यादा से diluted concentration नहीं रखते हैं. So whenever we do DNA purification or RNA purification, the maximum volume what we go is from 20 to 50 microliter only. So from your 20 to 50 microliter, like whatever the kit you are using for your DNA purification, depending on that, either you will have 20, 25, 30 or 50 microliter. इससे ज़्यादा नहीं जाते, इससे कम भी नहीं जाते. But if you think that during your sample preparation या DNA lose हुआ है, so what can you do guys is that instead of adding 20 microliter at the end, या फिर आप किसी बफर में आप उसको elute out करोगे या किसी deionized water में करोगे. So if you think that during the preparation your sample got lost, so what you can do is instead of 20 I can keep it as 10 microliter. ठीक है, so 10 microliter में the concentration will increase. Now what I'll do is from this 10 microliter I'll take 1 microliter of sample solution, I'll put into the nanodrop and check for its concentration. The concentration मुझे इतना कुछ आ रहा है. Okay, let us say that I got this much of the concentration. Now I can back calculate. So 1 microliter में इतना है, तो of course मेरा 10 microliter में कितना होगा? Around 5.6 microgram, can I say? Yes, है ना? It is the same thing, right? If I write it like this, 5.6 microgram or 5,600 nanogram. It is the same thing. Okay, so this back calculation you can do. Guys, are you getting the idea?
Yes, ma'am.
Okay, so now this is how you actually going to calculate. Okay, anyways, we'll do much of this when we are going to talk about methods in biology, which I will be starting soon. Okay, now what is happening is that coming back to the DNA damage, in the UV light, it is going to affect the DNA. Now, when you expose DNA for longer duration, अब जब हम expose करते हैं, we are going to, if I'm using a nano drop, then it is only 1 microliter of solution जो मैं use करके फेंक दूंगी. Of course, I cannot take it back 1 microliter मैं वापस नहीं ले पाऊंगी. It has been used, okay? So that's how it is actually saving a sample instead of putting 1 ml and all. Now, if I expose DNA for longer duration to the UV light, okay? The one problem is that a photochemical fusion of two pyrimidines will happen, and that is especially happening in the case of the thymine. So you must have heard of it. What is a photochemical fusion which results in the two thymine? Then that results in the formation of the thymine dimer. I hope you must have heard about it. Thymine dimer, which is also
Yes,
one of
ma'am.
the cause of your skin cancer. Yes, सुना हुआ लग रहा है कुछ?
Yes, ma'am. A covalent bond between the thymines, right?
Yes. Oh. Yeah, sorry about that. So this is how it is going to form. So you have got the DNA. यहाँ पे ये एक strand दिखाया हुआ है, ठीक है? So you can see that the bases are there. Now when you have continuous exposure of your UV light, okay, and especially your near UV, okay. So we categorize UV in two ways, near UV and far UV. That is nothing but just about the wavelength. So if I say that 200 to 400 is the UV range, right? 400 to 700 is the visible range. So 200 to 400 is your UV range. So near UV क्या होगा और far UV क्या होगा यहाँ पे? From 200 to 400 nanometer, if I'm asking what is near UV and what is far UV, where it is going to come? Far UV means UV से दूर, यानी कि visible की तरफ जाना. Okay? So इसका मतलब क्या हुआ? This far UV is going to fall in this area, whereas near UV is going to fall in this area. Okay, so usually we say that from 190 to 320 nanometer. ये range में हम near UV हम check करते हैं. ओह शायद गलत बोल दिया क्या मैंने? Hello, you guys are there?
Yes, ma'am. Yes, ma'am.
So this is yes, this is सॉरी मैंने उल्टा बोल दिया. नियर यूवी आपका ये है, सॉरी यस नियर यूवी इज दिस एंड फार यूवी इज दिस. ओके, आई जस्ट गॉट कंफ्यूज्ड. आई हैव डन द एक्सपेरिमेंट्स आल्सो, तभी मुझे डाउट हुआ कि मैं कुछ तो गलत बोल रही हूँ. एनीवेज आई विल से वंस अगेन. सो यू हैव डिवाइडेड यूवी रेंज इनटू फार यूवी एंड नियर यूवी. ठीक है, ओके. सो फार यूवी इज कमिंग, मिडिल रेंज क्या हो गया आपका? अराउंड 200 हो गया है, ठीक है. देन द शॉर्टर वेवलेंथ, जितना ज्यादा शॉर्टर होगा वो आपका फार यूवी में आ जाएगा. तो फार यूवी इज अराउंड योर 190 के बीच में आ जाएगा, अराउंड 190 नैनोमीटर. नियर यूवी आ जाएगा आपका 320 से लेके 400. बीच का हो जाएगा आपका मिडिल यूवी रेंज, विच इज लाइक इट कैन फॉल और इट कैन ओवरलैप बिटवीन द फार एंड नियर यूवी. इज दिस मच इज क्लियर टू यू? ये रेंज आपको क्लियर है? This is like 190 to 250 nanometer. Clear hai? Because I need to tell you something more over here. Is this much clear?
Yes, ma'am.
Okay. Now, what you have to remember over here right now is that, like in future, it is going to help you. So far UV range, that is 190 to 250 nanometer, this range is used for your secondary structure prediction of protein. Okay. And near UV is used for your tertiary structure prediction of protein. Can you remember this much, guys? क्योंकि ये आगे जाके आएगा दोबारा. Okay, make a short note of it. I'll just repeat. So we use UV for protein structure characterizations, out of which far UV range is used for secondary structure, in which alpha, beta, turns comes. Alpha helides, beta sheets, and turns are used. Okay. Near UV में आपका tertiary structure आ जाएगा. So it will tell you whether it is globular or a barrel shape. किस तरह का आपका tertiary structure वो आपको पता चलेगा. Okay. So just make a side note of it. We'll come to that later. Okay. Yeah, so now what is happening? Back to the DNA damage. So when you have got continuous exposure of the UV light, this results in the formation of cyclobutane ring between the two pyrimidines, that is two thymine. So we can see the structure over here. So this ring is formed between the two dimers, okay, two thymine molecules. Okay. Now what is happening is that now in the case thymine is adjacent to cytosine, the resulting fusion is thymine cytosine addict, in which the thymine is linked via its carbon atom 6 to the 4 of cytosine. अगर दो हमारे पास thymine thymine है, then the cyclobutane ring is formed between 5th and 6th of the adjacent thymine. But if it's thymine and cytosine, then it is 6th and 4th of cytosine. Clear? Yeah. Now, what will happen because of this dimer formation? There will be a kink present in the DNA sequence, because of which the DNA polymerase cannot proceed further and it will follow from the DNA strand. आगे आपका replication नहीं हो पाएगा. Okay, so this is also a kind of DNA damage. Now, to repair this DNA damage, the pyrimidine dimer, what we can have is in the dark, there is another DNA photolyase protein which gets activated. Okay, so that will, come and look over here. So in the dark, it can go and bind, and whenever light falls, that light is used. Okay, that energy is used by the enzyme. That light energy is being used by the enzyme to break the covalent bonds which is found between the adjacent pyrimidines. Okay, and it is also known as simply the reversal of the photo damage, photo reactivation. So why it is called photo reactivation? Because the enzyme has used the light. Light ने ही damage किया था, light ने ही इसको repair किया है. Because the enzyme is making use of the light energy to break the that covalent bond which is found between the adjacent pyrimidines. Clear? याद रहेगा? Okay. Simple, right? Now, finally, we have got the gamma radiation and the X-rays, which are particularly hazardous. And I hope you know also that whenever जब भी कोई cancer patient treatment के लिए जाता है, so we always say chemotherapy and radiotherapy, right? So radiotherapy में which kind of rays we are using? It is the gamma rays we use, okay? So it depends on the doses of the gamma rays, like at what dose the person is getting exposed to the gamma radiation. Okay. Now the thing is that the person will be sitting in that chamber, एक room रहेगा जिसको हम लोग gamma radiation का chamber कहते हैं. Center में chair रहता है, they sit it over there and then they get exposed. Okay. हर एक जगह का अलग अलग होता है, and when I was at Tata Cancer Hospital, so I've seen like that. कि वहाँ पे एक room रहता है, chamber रहता है, the person sits over there for certain particular time and उतने minutes के लिए he or she gets exposed. Of course, they can't do anything. They simply sit over there. The thing is that it is painless, but the problem starts as the days are passing. So अगर कोई cell damage भी हुआ है, तो eventually it will be known with the passing days. Correct? अगर कोई effect भी हुआ है, cancer कम भी हुआ है, तो भी हमें वो आगे जाके ही पता चलेगा. Okay. So these are particularly hazardous because they cause double strand breaks in the DNA, which are difficult to repair. Okay. Now, जब हमें मालूम है कि it is causing double strand breaks in the DNA and it is difficult to repair, then why are we considering giving gamma radiation to the cancer people, cancer patient? Yes. Question clear है? अभी हमने बोला गामा रेज एक्स-रेज जो है आपका डबल स्ट्रैंड ब्रेक करता है डीएनए को, राइट? And which are difficult to repair. Double strand break means they are lethal to the cell. Okay, it is difficult to repair. We do have repair mechanism, but it is difficult. तो क्यों हम कैंसर पेशेंट्स को गामा रेज देते हैं? क्यों हम हमारे हड्डी का जब कुछ टूटता है तो हम एक्सरे से एक्सपोज होते हैं? तो हाउ मेनी टाइम्स, लाइक मेरी पूरी लाइफ में मैं भी 3-4 बार एक्सपोज चुकी हूँ व्हेनेवर आई हैड दिस बोन का इशू, राइट? Why still doctor is recommending to undergo treatment of gamma radiation? क्यों हम जब भी ऑर्थोपेडिक्स में जाते हैं तो जब भी कुछ प्रॉब्लम होती है बोन रिलेटेड, वी स्टिल गो अंडरगो एक्सरेज इवन दो वी नो इट इज नॉट गुड फॉर आवर हेल्थ, it's not good for ourselves.
But the amount of exposure is limited and low. They try to target as much as they can, and they do try to limit. And depending on the treatment, it is probably worth the cost.
Yes, so that is true in the case of X-rays. See, so we know that in a lifetime of a human, max to max 20 times he or she has to go under the X-ray radiation, right? Okay, so I agree. So we don't get that much exposure to the X-rays, and that's why we are safe. What about the cancer patients? They get high exposure. They get high dose of radiations at the same time. जो पूरे जो गामा रेस है जो हमें एटमॉस्फेयर से मिलता है दैट इज नेग्लिजिबल एज कंपेयर टू व्हाट दे आर गेटिंग सिटिंग इन द चैम्बर बिकॉज दे आर गेटिंग द प्योर फॉर्म ऑफ द रेडिएशंस. All other radiations are not present, only gamma radiation, and that too they get high doses like 5 gray हो गया 10 gray हो गया, they go to 20 gray also. Okay, so but why?
Again, I think that is worth the cost because otherwise they'll die if they don't get the treatment either.
ये तो फिर हम उनको और जल्दी मार रहे हैं, right?
Maybe they are targeting the organ kind of like where the cells are present, only focusing on that particular area.
Okay.
They do target.
Yes. So one
I've
point
seen
is
the
that
tattoo point thing that they do so that they can it helps target the radiation in one place.
So one point is that good. So one point is that of the entire body, wherever the cancer is, the main center or the tumor is present, they particularly target that area. Okay. Believing that, thinking that, or no, expecting that the cancer cells will die. Because the cancer cells, they don't put their energy in repairing the DNA. But we know that the normal cells, at least the normal cells will put the ATP, the energy or the effort in repairing the DNA. So it is expected or it is believed that the cancer cells will die because they are not going to put energy in repairing. They just believe in dividing. Okay. Whereas the normal cells will try to repair. So this is keeping this belief and this expectation, the gamma radiation is being exposed to the cancer patients. Okay. Good try. Good. That's the correct answer also. Okay. Okay, so ionizing radiation can directly attack the deoxyribose in the DNA backbone, or it can indirectly affect by generating the reactive oxygen species, so which we have just discussed, right? Reactive oxygen species, they can result in the formation of the oxo-G, which can result in the transversion. So GC ke jagah next round of replication, we are going to have TA. Okay, so transversion can happen again. It might lead to cancer. Okay. So guys, I'll tell you one more thing. Like since like now my company, I mean, we are working on cancer genomics, and you won't believe, I myself didn't know that there are so many types of cancer, like so many. Right now, the company has, made a table in which we have registered around 53,000 types of cancer. 53,000. This is which we were able to record. I don't know how many more are there. How many more? I have no idea. And especially in that, the सबसे ज़्यादा complicated cancer. Can you tell me? Can you guess which one it is?
Breast cancer.
Yes. The most complicated one is the breast cancer because first of all, okay, on what basis you said, then I'll tell my answer. Yes. On what basis you said?
Like I have read about breast cancer, like how fatal it is and how it is common among women. And I've read about the HDR2 overexpression also. On
You
that
have,
basis, I told.
you have read about what? I couldn't hear. What is it?
HDR2.
Yes. Very good. Yes. Correct. Very good. So in the breast cancer, okay, so you have got the varieties of cancer because of the DNA damage, right? In the case of cancer is that even hormones are playing a role in causing the breast cancer. And it is not only specific to the females, it is also for the male cases also. So in the females, the categories are more complicated. Like you have got the tubular, lobular is there, like wherever, sorry, one second. Okay, so wherever you have got those, what do you call those? Okay, what do you call that? It's a lobules, like the milk forming ducts are there. So, right. So because of that, you have got different categories, and what kind of the secretions it produces, you have got different kinds of that, and many more are there. Under this, because of the HER2, okay, because of this. So you can have hormone responsive, positive, negative based on that. Then you have got the HER2. This is human epidermal growth factor 2. Based on this, also positive and negative. You can have so many types of breast cancer. Oh my God. I just recently came to know about this, and I have even studied, but I never thought, the patients, are suffering from this much, and each category of patients are receiving different kinds of drugs. So it's actually very complicated, and I really wonder how beautifully the doctors are, able to detect this and prescribing the drugs to them. It's a huge task. And of course, like we are helping them. That's the job of our company also. But yes, so breast cancer is the most complicated. And after breast cancer, it comes the lungs. Again, in the lungs, several types are there. Several. Okay, anyways, so this is just the outside info. Okay. Now, because the cells require intact chromosomes to replicate the DNA, ionizing radiation is used therapeutically to kill the rapidly proliferating cells in the cancer treatment. Okay, so जैसे कि मैंने पहले बोला आपको कि cancer cells don't think about repairing. It always thinks about the replicating. They just want to divide. So it becomes easy for the ionizing radiation to kill those cells. Now we have also got the anti-cancer drugs such as bleomycin. It also causes breaks in the DNA. And ionizing radiations, agents like bleomycin, they are known as clastogenic. Clasto means breaking. So they are like, DNA breaking agents they are. So when nowadays, if you see, many people are thinking about target therapy, right? Target therapy. 1 second, guys. Okay. So I was telling about the target therapy. So when you say about the target therapy, like even I was working on this target therapy, I was working on pancreatic cancer cell lines. Okay, so I was working on cell lines only. I couldn't go further. So when I started working, like when you think about like how to do research, कैसे हम कोई drug market में ला सकते, it's such a huge process. You must have seen, like in the case of pandemic of this COVID-19 itself, so much research work was required, right? First, you should know like what kind of structure, how it is replicating, how we can get benefited, if we are able to prevent the spread. So you must have seen kitna time laga, but this is the first vaccine which has come into the market very soon. नहीं तो कम से कम 10 से 12 साल लगता है for any drug to come or any vaccine to come in the market. This is like first which has come so early. Can you give me the reason why this vaccine came so early in the market and it was accepted by the government, FDA and European medicine agencies also? Can you tell me why and how it was possible?
I read that they didn't go phase wise, like phase 1 के बाद phase 2 नहीं कर रहे थे. They were pulling multiple phases simultaneously.
Correct. That is one reason. And see, before going to the clinical trials on humans, like on human clinical trials, you do have to do a lot of wet lab work, right? But if you know, this corona is not new. It's not a new virus. It is actually a virus which is similar to our cold virus. Okay? So it is not new. So already हमारे पास इसका structure available था. इससे पहले SARS आया था. इससे पहले MERS MERS आया था. Do you know guys about this MERS and SARS? सुना वो लग रहा है? It is Middle East Respiratory Syndrome. It is severe acute respiratory syndrome जो I think China में ही हुआ था. हाँ, China में ही था. Again, yes. So
दोनों चाइना में तो है।
yes, yes. So already we knew the structure of these. Okay, these viruses, we knew the structure and it was known how to prevent them. ठीक है, जो भी anti-viral drugs है, it was known. तो जब कोरोना आया, so they found that it is belonging to the same family. अब same family में बस एक दो amino acid इधर उधर होंगे, but structurally it is going to be similar, right? So that's why it was easy. अभी हमारे पास इतना technology है, इतने computer simulations हैं, so it was easy to get the structure. जब हमारे पास structure है, तो हमारे लिए ड्रग या एंटीवायरल ड्रग बनाना इजी हो जाता है. ओके, बट वैक्सीन बनाना था, इतने सारी जनता को ड्रग देना पॉसिबल नहीं था, क्योंकि इट वाज टेकिंग टाइम. सास फिर भी थोड़ा सा सीवियर था. कोरोना इज लाइक लिटिल माइल्ड. इफ यू आर इन हेल्थी एज तो आपके लिए इजी था टू रिकवर करना. यू मस्ट हैव नोन, राइट, कि जो बूढ़े लोग थे, सो मेनी डेथ केसेस वे देयर इन दोज एज रेंज में, है ना. So if they go till ventilator and सांस लेने की problem, if it has affected the lungs, then it was difficult for them to come back. Yeah. Now, if they were in the middle age, like 30, 40, it was possible for them to come back. ठीक होके दवाई ववाई लेके. Now, vaccine, if you know, India ने क्या किया? They have used attenuated vaccine of the entire coronavirus. Whereas outside the Covishield है, they have used the mRNA based. Okay, CRISPR-Cas9 method उन्होंने use किया है, and that's how they got the vaccine. So इसलिए Indians, they said that, you know, it is going to help you in giving the immunity. It is not going to prevent you from getting the corona. Correct? If you remember. So still we need to wear mask and हमें precautions लेने हैं, because still you can get affected. It's just that you will get immunity to fight to that. Okay. Now, same thing, target therapy when I was talking about. So when you say target therapy means what? आपको पता है कि किस जगह पे मुझे कैसे टारगेट करना है. Now, for example, pancreatic cancer cell lines पे I was working. So I was targeting the receptors which is present on the pancreatic cancer cell lines, which were specific to the cancer cell only, that too of pancreas. Okay. So I did get my drug which was working and all, but later I came to know that if it is injected into the blood system, it was losing its activity. And that's why, of course, I couldn't work further. So आपको research करते टाइम पे पता चलेगा. So when you do target therapy, how difficult it is. Okay. ये बहुत simple रहता है. Like bleomycin, just add in the system, जहाँ पे भी जो जैसा DNA मिलेगा, they are just going to be breaking the DNA and causing the cells to undergo apoptosis. So along with the cancer cells, they were also killing the normal cells, and that's why we don't use such drugs now. Okay, getting the point? Nowadays, we go for the personalized therapy. अभी target therapy से भी दूसरा शब्द आ गया है, that is known as personalized. Now, what do you mean by personalized therapy? Yes?
It is individually designed for the particular person.
Yes, particular patient. Okay. So अगर आपको इस particular type का cancer है, like I was telling you, around 53k types of cancers are there. Now suppose you have got कौन सा वाला बोलूं, like hormone receptor positive HER2 negative breast cancer, ठीक है? तो इस तरफ का है, so then you are going to have a specific kind of drug for this. Okay. Now, what kind of age you are falling into it? अगर आप छोटे हो, middle age में हो, adult में हो, old age में हो, then again this classification varies. ठीक है? So इस हिसाब से, you know, for particularly for that particular patient, the drug regime is being now prescribed by the doctor. Okay, but अभी ये सब बाहर ही हो रहा है, US में and Europe में they have just started it. इंडिया में इट इज लाइक जनरलाइज नाउ. अगर आपको लंग कैंसर है तो मतलब वो पूरी कैटेगरी को लंग कैंसर का ड्रग मिलेगा. ओके, सो दे हैव नॉट कैटेगराइज लंग कैंसर इनटू कौन सा वाला टाइप का. या बट पीपल आर देयर वर्किंग ऑन दैट and we'll soon reach that stage where we can also give personalized therapy in India. Okay.